So welcome everyone. Yeah, it's my distinct honor to introduce our distinguished guest, Professor Robert Leinhardt. He received his PhD from Vanderbilt University and is currently an associate professor in the Department of Psychological and Brain Sciences and Biomedical Engineering. He is one of the world leading scientists in the cognitive neuroscience, particularly in non-invasive neuromodulation. He has published his work in the most prestigious journals, such as Nature Neuroscience, Science, Nature Medicine, and so on. He also received Science and Peace Prize for Neuromodulation from AAAS and the Innovator Award from International OCD Foundation. Yeah. He will give us a talk titled with From Coordination to Control, Targeting Neural Rhythms to Modulate Brain Circuits. Without further ado, please join me in welcoming Professor Robert Leinhardt with warm applause.
Okay, well, thanks for coming to the talk. So just by way of introduction, I want to thank my sponsors up front, NIH, private foundations, philanthropy, and also highlight two postdocs in the lab, Wen and Shrey Grover, whose work I'll mainly be presenting today. They headed up these projects and are primarily responsible for making the major contributions to them. I have nothing to disclose, basic researcher, like many of you. So I want to start by highlighting a central question I think that many of us are interested in the neurosciences, as to whether we can move beyond simple observation of phenomena and actually start controlling cognition and behavior. If behavior, as we think it is, emerges from coordinated brain activity across circuits, then the problem is not just where in the brain activity occurs, but how it's coordinated. A growing body of work over the decades has suggested that this coordination, this communication in the brain, is subserved by brain oscillations or brain rhythms. And so the question that I'd like to ask today is a pretty simple one. It's that can we control thoughts and behavior by tuning brain rhythms? And for kind of basic scientific discovery, as well as for potentially translation, for potentially fixing brain circuits when they go awry in neuropsychiatric and neurodegenerative diseases. So again, what I want to suggest is that it's not that the stimulation site is the only thing that's important, but the kind of coordination code in the brain. And there's different ways of targeting these codes non-invasively and safely using causal neuromodulation tools, non-invasive brain stimulation tools. They come in various flavors, right? There's electrical brain stimulation, there's ultrasound, there's TMS. Within these classes of tools, I want to focus first on one particular tool called high definition transcranial alternating current stimulation, HDTACS. And HDTACS is a low intensity electrical brain stimulation approach, a neuromodulation approach that allows us to safely, non-invasively inject electrical fields into the brain that can interact with ongoing brain activity. The reason why we like this tool is not just because it has spatial targeting ability, but again, it can affect that kind of brain communication code that we think is important. So it gives us essentially three degrees of control that I want to highlight. One is frequency control, that is, what rhythm are we targeting? Phase control, right? Like how regions are coordinated across space. There are these timing relationships or phase relationships. And then the third is this standard one that you get with most brain stimulation tools, namely anatomical targeting. Where in the brain do we want to intervene? What circuit, what node in the network? So again, rather than stimulating a particular region, I want to underscore the cool thing about this tool is that we can target, we can start thinking about targeting dynamics of brain communication. So a snappy phrase is, modulating codes instead of nodes. That will be the only joke today. So here's how I've organized this talk. It's a little unusual. Usually I present like a small subset of studies we do in the lab and do a deep dive into those studies. Today is going to be very different. I'm going to give like a chef's tour through a lot of terrain, a lot of different studies to try to draw some high-level mechanistic insights. So I think it will be interesting for all of us that way. But by definition, there will be a lot of loose ends to tie up. So most of the work is published. You can check it out. We can talk during Q&A, shoot me an email. But there'll be these methods gaps if that's intentional, or just a byproduct of trying to cover a lot of terrain. So the first thing I'd like to do is discuss brain rhythms as control variables for behavior. And I'd like to try to convince you that we can use tools like TACS to manipulate these variables causally, first in cognition and then across memory and control domains of processing. Then I'd like to show you how we've applied this logic to symptom-relevant circuits for psychiatry. I'll also make a note about neurodegenerative disease briefly. And then finally, I want to dawdle over some of our latest work that I find most interesting because it's fresh and new. It essentially is extending this logic, this framework, into deeper brain structures. So there's the roadmap. Okay, so first, we've investigated this question of whether oscillatory synchronization is a causal control variable. That's the claim. So let me show you a series of studies where I think that supports this claim. So here's a study from 2017 where we've devised this HDTACS protocol targeting the medial frontal cortex and the lateral prefrontal cortex. This is a bit unique. Usually you stimulate a region. This is stimulating two regions simultaneously using HDTACS. And we're particularly trying to target a stream of connectivity across these regions. Because this connectivity has been very well characterized across over 3 decades of animal, mainly monkey and human research as a network signature of executive functioning. So it's nice. We're riding on the heels of 3 decades of research that have characterized this very extensively. So we know it has a strong functional and behavioral significance. What's novel here is we're trying to target it causally using HDTACS. It's a connectivity target, which is a bit novel for using TACS, not just a single region. And we can tune the TACS to apply a 0 phase degree offset between these regions. This is sometimes called a synchronizing approach or in-phase stimulation. The idea is, conceptually, we're trying to facilitate the neural integration between these regions. And we can also flip the phase angle, make it 180 degrees phase offset across these regions, apply what's called an anti-phase montage with the goal of destructively interfering with this communication across these regions. So if you're not familiar with this type of cognitive neuroscience, I'll make it simpler. So if you make a mistake on a task, you have this like alarm bell goes off in your brain. Lots of things happen, but the mid-dorsal anterior cingulate cortex fires a lot, and that activity is readable over the vertex of your scalp right here. Extracranially, where you put a little electrode there with some gel, and you can read that very quickly after you make a mistake on a task. It's like your brain saying, "Oh shoot, something's wrong. I need to make an adjustment. I need to make a change." That signal's been, again, studied to death. So we know it's there in lots of different contexts and species and so on. And then that same signal is communicated to various parts of the brain. One in particular is the dorsolateral prefrontal cortex to do various things, essentially like implement the control. So the alarm bell goes off saying, you need to pay attention better. You need to increase the need for control over your thoughts and behavior. And then it sends a signal to DLPFC being like, implement that control, update a goal representation, whatever. Okay, that's the basic sketch. And we want to we want to reach into the brain, latch onto this stream of communication between these regions, and see if we can change it. So that's the montage, very briefly. Montage is sort of where we put the electrodes, where the intensity of the electrodes are, what we're targeting. Here's the task people are performing. It's a time estimation learning task for the cognitive human neuroscientists using EEG aficionados in the field. This is the classic Miltner et al. 1997 experimental task that they used to discover the feedback-related negativity. If you don't know what that means, that's totally cool. But this is a time estimation learning task. So you're estimating a time interval, very simple. Comes in like this as fixation. There's a target. You make a button press response after some time has passed. And then you get feedback about whether you should speed up, you should slow down, or you should stay right where you're at. So you're trying to estimate a time interval with a button press. Now the wrinkle is there's feedback, there's blocks of feedback trials, there's blocks of non-feedback trials. And as you'll see in a second, that's nice because we can start to assay the memory representation of that time interval over time and quantify it using like a block by time interaction. We can also evaluate adaptive or executive control. How will you adjust your behavior after you receive punishing feedback on this task? Again, that was this is the original discovery of the feedback-related negativity, where this was discovered decades ago. So we're going to use the same canonical task. Subjects, this is everything I'm to discuss today is kind of the gold standard in the field, double-blind, sham-controlled, randomized studies. Most are crossover, healthy younger adults. If not, I'll mention they're between subjects or they're with other subject populations. Subjects are performing this task while they receive brain stimulation, in-phase, anti-phase, or sham. Sham is just you turn the stimulator on, you get that itching, tingling, poking sensation for the first 30 seconds, then it ramps down, and it ramps on and off at the end of the 20-minute stimulation session to simulate that itching and tingling sensation that people endorse on active testing days. So it's our kind of the field's gold standard. But you'll see in subsequent experiments, we kind of go beyond and we do these platinum standard, like the more active controls in addition to this more passive control, but this is the standard approach. And you see people come in on 3 different test days as crossover within subjects. They come in on 3 different test days. They receive these stimulation conditions across those 3 days. Order of stimulations counterbalanced across subjects, and and most of the analysis is going to focus on these after effects after the stimulation window has stopped. So this period post-stimulation is where we do the EEG analysis. So everything I'm showing you is post-stimulation. So there's no TACS artifact to be concerned about. So here's one of the major findings neurally is this bidirectional causal control of theta synchronization across the medial lateral circuit and frontal cortex. So again, over here, the vertex of your scalp, nearest medial frontal cortex, and over here, right DLPFC, there's a stream of low-frequency oscillatory phase synchronization. That just means fast neuronal correlations across these regions. It's an aspect of brain connectivity. That's observable, replicating, again, like 3 decades of research right here in the sham condition, the center time-frequency representation. And you see it's very, this is the point, 0 is the point at which there's negative punishing feedback on this task relative to positive feedback. That's a difference map. So it isolates this, this adaptive control response. You see it's in the theta band, roughly 4 to 8 hertz. It appears pretty fast after the onset of negative feedback versus positive feedback. Here's a seated connectivity analysis in source space. If you're interested in spatial aspects of this profile. One thing you'll note, I'll show you later, is that these blobs are more or less the same over right hemisphere, left hemisphere. But with the stimulation conditions, there's a preferential impact just on the right hemisphere where the stimulation was applied. And that's the major finding here. When we apply the antiphase stimulation, we get a downregulation of this connectivity. When we apply in-phase stimulation, we see upregulation of this connectivity in a spatially specific manner. The behavior also changes in the expected way, where learning and executive control measures are enhanced with in-phase stimulation when this network signature is enhanced, and it's deficient, it's reduced when we reduce the connectivity in these regions. So here you're looking at measures of learning. This mean error on the top is a measure of learning. Mean variability is also a measure of learning. And then down here, adjustment efficiency. That's how well you adjusted your behavior after you made a mistake on this task. That's a very well-known measure of executive control or adaptive control. To orient you, here are the blocks on the x-axis. And you see the feedback blocks are in gray, and the non-feedback blocks are in white. So you start with no feedback on this task. And then when you receive feedback, you do better. You adjust your behavior better, you learn faster. And then we're gonna take that feedback away. And over time, you can see the learning happen as a block by time interaction over the course of the recording period. So you can see how the line flattens out. And that's evidence of learning. You don't need any more feedback, and you're still responding very consistently. So I hope you can appreciate. Okay. I hope you can appreciate that the antiphase stimulation is red here. You see worse learning. You see impaired executive control. And the blue is in phase. You see enhancement in learning and executive control. In a follow-up experiment, we took this one step further, and we induced deficits, and then we rescued them with the in-phase stimulation. So here you can see the sham condition, where we get baseline levels of people learning and executive control behavior. The red is the antiphase, the destructive interfering stimulation. We can induce deficits, just like we showed previously. And then we can apply the in-phase stimulation, same individuals, and rescue that executive functioning behavior within minutes. It's not just a passage of time effect, that we can do the same thing and just let time continue without applying the in-phase beneficial stimulation, and people remain in deficit. We next asked how constrained this effect is mechanistically. So first we see a high degree of frequency, spatial specificity. So here it's just the theta band connectivity that shows the bidirectional effect. In phase improve, anti-phase inhibit, not delta, not alpha, not beta. It's just the right hemisphere that's showing the effect here, not the left hemisphere. And spectral power within these sensors of interest are also not changing. We're changing, it appears, timing relationships across these regions, not the sheer intensity of the neural activity within these regions. So again, everything remains the same here in this experiment. Same intensity, same subjects, same frequency, same locations, everything is the same. We just flip the phase angle of the stimulation. You flip the phase angle, the whole system flips with it. There's also a degree of frequency specificity in the behavior. You apply a 35 hertz gamma band TACS in the same manner to the same individuals, no change. The bidirectional effects were also apparent at the single trial level. So the beautiful thing about this neural effect is it has such high signal to noise and it's functionally relevant to next trial slowing behavior. Right, the canonical measure of the implementation of executive control. And if you compute phase angle differences within a trial across regions, you can get this measure of connectivity within a single trial. And you can correlate it with next trial behavior, slowing. The stronger that neural connectivity signal is, the more slowing you get. That's been shown for a long time now, so this is just replication right here. The functional significance of right lateralized phase connectivity is stronger than the left hemisphere connectivity. That's also been known for a while. The novel finding here is this bidirectional control at the single trial level. We see the same thing at the meta-analytic level. Recent meta-analysis that we performed, the largest to date, over 100 TACS studies on cognitive measures. Here, executive control, working memory, various cognitive measures, just like a big picture snapshot. In phase improve, anti-phase inhibit. The claim, again, is that we think oscillatory phase is a causal control variable to pay attention to. We're following up on this study with another series of experiments to try to extend this phase-based control using repeated and personalized brain stimulation. So here, we want to repeat the stimulation over numerous days to see if we can extend beyond the minutes time scale after effects that I just showed you to longer time scale in durable post-stimulation effects. We're also personalizing the stimulation to personalized theta frequencies using the network signature, and we're also broadening to see if the effects generalize to other similar tasks, so not just that one task. What we see is the same phase-dependent effects. Here we have an interest in impulsivity. So this is the Brett impulsivity scale, very known self-report measure of impulsivity in the field of impulsivity. And you can see that in phase reduces impulsivity, anti-phase increases impulsivity, makes you more impulsive in these healthy college students from the greater Boston area. So you get this nice bidirectional change in impulsivity. We can make you more impulsive or less impulsive just based on the phase angle of the TACS that we're pushing and pulling through this circuit. It also, we also observe the same bidirectional causal changes in other tasks. A go, no-go task, looking at impulsive action. A time estimation learning task, looking at impulsive choice, early responses. And again, in this extension and replication study, which is on these bottom panels, again, the BIS changing bidirectionally. So again, this kind of raises that possibility that synchrony acts as a causal control parameter here now as potentially like long-lasting state change. Another question we were curious about is whether these same principles, frequency, specificity, phase, specificity, extend to other domains like working memory, which is of great interest to us as basic researchers, but also is notably impaired in many neuropsychiatric illnesses. So here in this task, in this working memory series of experiments, we're using this visual working memory delayed match to sample task with complex real-world objects.
Here's the task. You fixate, you get a complex real-world object. There's a delay period for 3 seconds. You see another object. Just indicate with a button press whether it's the same or different from the original. This is the memory trial. Then there's like this slick control trial manipulation where it's identical to the memory trial, except the task is here at pro. So you indicate with a button press whether this grading is tilted left or right from the vertical, and it's followed by a mask. So what's really lovely about this design is that it allows us to isolate working memory neural activity independent of stimulus evoked activity. The blocks are titrated to accuracy, the vigilance, attention, control. While participants are performing this task, this is a RCT in healthy younger and older people. While they're performing this task, we're applying brain stimulation to frontal temporal regions. Again, 2 regions concurrently to try to isolate phase synchronization between these 2 regions. There's a big backstory here of literature. I'm going to skip over it, but you can trust me that these 2 regions are importantly implicated in the maintenance of information as you carry forward in time in your mind's eye. And this is phase specific. So we're doing the same in phase, integrate or antiphase, destructively interfere. It's personalized to each individual's peak theta frequency of the long-range phase synchronization across these regions. So the connectivity has a frequency component to it. We can measure that using EEG. We can find the peak frequency, and that's the frequency we apply to your brain when you come in for this experiment. When we run this test in older people, they have deficiencies in long-range functional connectivity between these frontal and temporal regions, and absent theta gamma phase amplitude coupling in the left temporal cortex. Theta gamma phase amplitude coupling, theta phase synchronization, these are probably the most, arguably the best well-studied indices of neural plasticity and communication in the brain, particularly how they relate to memory processing. And the major finding here is that when we attend to the features of this circuitry and design stimulation that's key to the spatial spectral features of this circuit, we can manipulate the circuit causally with brain stimulation. And we can improve in older people this long-range, low-frequency connectivity, as well as this theta gamma, this kind of nested oscillatory signature in the temporal cortex. For those not familiar with this, when you store information in your mind's eye, the perceptual features are being coded by neurons in the back of your head, essentially. Like here, like IT cortex is like the top of the cortical hierarchy, important for coding complex objects, real-world objects. And then the prefrontal region is important in kind of reaching back like a fulcrum and controlling those neurons and keeping them online. So it's important for like monitoring and maintaining and top-down control. That's the conceptualization that's applied here. When we improve these neural signatures in healthy older people, we also get benefits to visual working memory performance, accuracy, success on this task, but not reaction time. So we can make you better with brain stimulation, but not slow you down. It's also suggestive it's not a simple speed accuracy trade-off. You didn't just slow your performance down to make more accurate responses. You're getting better without slowing down. That's a kind of preferential impact of stimulation on working memory success in older people. Subsequent experiments, we also show the same bidirectional causal control. We can apply the antiphase version of this montage in healthy young people and see working memory deficits. We've run a series of experiments to replicate, extend this. I want to show you one of them that I think it's particularly rigorous. If you're in the field, you're familiar with brain stimulation field, no matter what the tool, if you want to try to establish frequency specificity, you typically look at neighboring frequencies, or some frequency that's like atheoretical, right? Spatially, you would go to another spatial location on the brain if you're trying to establish spatial specificity. If you're testing that, it's a common question to do. You might go to motor cortex. That's very common. What's kind of unique, like rigorous about this is, as you'll see, the frequency is within the frequency of interest that we're going to test, just not personalized. And spatially, it's within the spatial regions of interest, just not applied concurrently. So here we want to, in this new cohort of older people, RCT, crossover, they come on 5 separate test days. They receive the baseline sham, kind of classic passive sham. The frontal temporal in-phase tube is like personalized. So this is what I showed you previously. And then here are the 3 novel extension simulation conditions. Frontal temporal in-phase non-tuned, so not personalized. So they're just going to get a uniform 8 hertz frequency applied, so not frequency personalized within the theta band. Frontal tune, so personalized, but it's just the frontal region alone, and then temporal region alone. This is what we found. We reproduced the principal behavioral finding of a benefit to the visual working memory accuracy without slowing people down in terms of their reaction time. And we see no effect of the other conditions. So it looked like, at least for this type of working memory function, it's sufficient to engage the circuit concurrently, frontal temporal, and to do so in a manner that's personalized to the physiological frequency of the circuit. We're now testing this protocol in people with mild cognitive impairment and mild Alzheimer's disease. We have funding to do this. It's just some pilot data that supported our ability to get this grant. The same task, we just have a set size manipulation of 2, 4, 6 different objects that would require you hold in memory. And this N of 10, small sample size, shows that in healthy older people, you see the classic improvement in memory capacity until it hits an asymptote. People with MCI-AD just have this decline across memory load, but with brain stimulation, we can go some way in improving these patients and making them look like the healthy older individuals. And we also see an effect in theta gamma phase amplitude coupling after stimulation. So more on that in the future, some kind of titillating early results. The next question is whether these same principles extend across different memory systems. So here we turn to the classic serial position curve effects, which I'm sure you're familiar with. This has been studied in experimental psychology for literally 100 years. So if I read you a list of words and you recall back to me as many words as you can from that list, you'll have a behavioral advantage for the words early on that list, deemed primacy, which is linked to long-term memory and the classic dual store framework. And you have a behavioral advantage for words later on that list, deemed recency, related to working memory. Because the effect has been studied so long, we know quite a bit about the functional neuroanatomy underlying recency and primacy serial position curve effects. For example, that parietal lobule is a major player in recency, and dorsolateral prefrontal cortex, a major contributor to primacy. What we don't know yet is whether there are actually rhythmic mechanisms within these segregated cortical regions, DL, PFC, and IPL. But if there are, then we could potentially use tools like HD-TACS to preferentially independently modulate these memory systems. Again, because TACS has the anatomical targeting to approach this question, and it affords frequencies control. So to test this, we ran a series of RCTs in older people, 150 older adults, age 65 to 88. We wanted to see whether we can, induce durable effects. So we again stimulate repeatedly across several days, and evaluate memory performance before the intervention, every day of this 4-day intervention, and 1 month after the intervention. We hypothesized that in training, theta oscillations in parietal cortex would change recency, working memory, auditory verbal working memory, and in training the higher frequency oscillations of prefrontal cortex would preferentially impact long-term memory primacy. So just to orient you, here's what the data looks like. So probability of recall on the y-axis, primacy, 3 middles, and recency on the x-axis. This is the classic kind of serial position curve effect. So you see advantage early on the list, later on the list, later on the list. So there's 2 different experiments. It will become obvious what experiment 2 is in a few slides. Essentially, we just switch the entrainment frequencies across the locations to test the frequency and location specificity. The major finding here is that when we entrain gamma oscillations in the DLPSC, we see a change preferentially in primacy, long-term memory, starting on day 2, day 3, day 4, continuing to the 1-month time point, above and beyond that of the baseline. That's preferential, so it's not a change in recency, and nor does state oscillations in the prefrontal cortex have any change but minus your recency. Conversely, when we entrain low frequency theta oscillations in private vortex, we see preferential enhancement on day 3, day 4, and 1 month post. Not primacy, and not when we use the higher frequency stimulation. No effect on primacy or recency. Just notice that were we to just do a one-off, a one-shot stimulation, we would have observed all null results, maybe concluded stimulation doesn't work. It didn't work after 1 day or 2 days. We had to keep going with the stimulation to see the effects emerge, and they sustained well beyond the stimulation window. 1 month is quite a long time for these types of studies. We see a double dissociation between location and frequency on the one hand and a memory function being modulated on the other hand. What's not, now if you're familiar with this literature, we already knew the double dissociation in terms of space. Frequency piece is new. What was also interesting is you look at these big intervention studies and typically you see pre-post assays of brain activity, behavior, other things. You don't often see measurement during the intervention itself, but we did that. So we had a kind of a unique opportunity here to look at how things are changing during the intervention. And what we're finding is that just a simple linear modeling of memory enhancements over these 4 days, you take the rate of that enhancement and it predicts how big your memory benefit is 1 month later. So that was pretty cool. Obviously some potential clinical utility. We're talking here about stimulation responsiveness. What predicts it? You can imagine maybe into the future, you're a patient in the clinic, the doctor gives you stimulation. How you're responding to this stimulation over the routine prevention is going to give us information about how well you're going to do 1 month later or in the future. But that's speculative. Another cool thing was if you look at another important variable in determining stimulation response was how people were when they came into the study at baseline. We looked at general cognitive performance measured by the MoCA, the Montreal Cognitive Assessment. And if you came into the lab before the study starts and you're a very low general cognitive function person, you benefit the most during the intervention and 1 month later. That's the upshot. We also see some encouraging signs in the literature as a whole. Here's the meta-analysis again. We're able to cleave off just the TACS cognition studies done in healthy older people that had a sham control. And we see small to medium size effects in the literature as a whole as of 2023 when we did this. One thing to note is that we think this undersells the potential of tools like TACS. These are conventional TACS studies. So big sponge pads, bad spatial targeting, no personalization, no repeated stimulation. They're not targeting oscillatory phase angles across regions. This is kind of vanilla TACS, but we're still seeing small to medium size effects. So it's encouraging, but we think there's a lot more potential here. That's our cautious but optimistic assessment. Again, I want to continue with this chef's tour and just touch on a lot of studies. So now I want to tell you about how we've been thinking about how these principles might extend to more clinical domains, extend to maladaptive behaviors. And we've been particularly interested in compulsive behavior. And so for this study, we combined insights from 2 disparate literatures. I had the good fortune of being in psychiatric neuroscience for a while before taking this professor job, particularly in schizophrenia labs. And so it was nice to have kind of a hand in that literature and that being trained in cognitive neuroscience, because you can look at literatures that like these two that rarely make any contact, but actually come together in some interesting ways potentially. And that's what this exemplifies. So in psychiatry, it's a large literature on functional neuroanatomy of OCD, obsessive compulsive disorder, and that literature shows that compulsivity is linked to dysfunction in orbitofrontal striatal circuits, particularly during reward processing. And in cognitive neuroscience, that's developing in parallel, but again, is not talking to this biological psychiatry literature, has shown that reward processing is associated with a frontal beta gamma oscillatory response. So in a learning or decision-making context, when you get a positive outcome, the overall frontal cortex neurons fire, some other regions too, like ACC, and you get this signal, again, over the vertex of the scalp in this beta gamma frequency range, like 20 to 30 hertz. And this is, again, from animal research thought to arise from oral frontal cortex. So, I mean, we surmise that maybe this beta gamma rhythm in oral frontal cortex could be a control signal for reward guided behavior. So that's what we set out to test using HDTACS, targeting oral frontal cortex, using a monetary reinforcement learning task that separates reward-driven learning from punishment-driven learning. And we had subjects perform this task. We measure the reward beta gamma feedback effect. We find your peak frequency, and that's again personalized. So this is personalized beta gamma frequencies targeting oral frontal cortex with HDTACS. And behavioral study, while subjects perform this task, we look at online behavior, and we see a selected change in reward-driven learning, here blue. This is optimal choice over trial numbers on the x-axis. He selected change in reward-driven learning, no change in punishment-driven learning in the yellow, and no changes in either type of learning in the passive sham control or the active control at a alpha frequency, 10 hertz. Actor-critic modeling of behavior showed that the stimulation had a preferential effect on increasing exploratory choice, the beta temperature parameter, which is consistent with modulation of the underlying learning policy. We then went further in a subsequent experiment, and we asked, well, like, given that compulsivity has been previously linked to excessive reward habit learning, and given that we have potentially a tool in our hand that can down-regulate reward-driven learning, maybe we can apply it to people with obsessive compulsive behaviors and see attenuation in those types of behaviors. So that's what we did. This is another double-blind RCT. Here we're enrolling individuals with elevated obsessive compulsive tendencies, obsessive compulsive traits, but without a formal diagnosis. So this is not OCD patients, this is subsyndromal. But we're finding using the obsessive compulsive inventory revised assessment is that that we can cut obsessive compulsive tendencies in this subclinical population by a third. And by delivering this personalized brain stimulation over 5 consecutive days with effects that last for 3 months. We also find, again, that these baseline variables matter, just like we saw previously. So you come into the lab with really high level of compulsivity behaviors. We can't call them symptoms, but these types of tendencies, and you're going to benefit the most from the simulation. You're going to see the largest reduction in those tendencies after the 5-day intervention. So it's kind of cool. The pattern that emerges, it looked promising. We have a kind of mechanistically defined signal. We're targeting, we think, is the right circuit. And we're producing durable, behaviorally meaningful change. That said, of course, this is subsyndromal, so we don't know if it's really going to work in OCD. Will it translate to clinical OCD is the question, and that's what we're pursuing now in the lab. So currently we're running RCT and OCD patients in collaboration with our clinical partners using the same protocol. So this clinical trial is ongoing now. I don't have data to show you, but it was motivated by the work I just showed you in the subsyndromal case. It was also motivated by some kind of cool, like, N of 1 case studies that I'll show you. So I don't want to overinterpret a small N, but I think it's still kind of cool. So this is someone that is treatment resistant, OCD patient. It's just like, it's 4 decades. This person's been suffering. They've tried electroshock therapy, TMS, drugs, everything. They're not getting better. And we bring them in through our 5-day intervention. We see a radical reduction in Y-box scores. This is the classic symptom measurement in OCD. So this got us really excited, and our funders as well. And so this was part of the motivation for the clinical trial we're running now. I showed this, and I gave this talk of biological psychiatry in New York City a few months ago, and someone came up from the audience and saying, oh, we're seeing something very similar, treatment-resistant OCD, intracranial recordings, and this same like beta gamma signature in the OFC. So we're proposing it might be a novel biomarker and treatment alternative for OCD. And this was our work in 2021, but just last month, there was a paper in Cell by Casey Halpern's group at UPenn, converging on the same conclusion for radically different approach using again, another group now, using intracranial recordings in chronic severe OCD patients. In this case now applying electrical brain stimulation to the orbitofrontal cortex and seeing dramatic treatment relief, symptom relief. And so I got in touch with Casey right away. Actually, our funders put us together, and we're writing a commentary now on this potential novel biomarker and treatment for OCD. I think it's pretty cool that we had no kind of knowledge of each other, and we arrived at the same conclusion. But the problem is not just access, but causal selective access at the level of computation. And so there's this newly introduced tool called temporal interference stimulation. It involves kilohertz electric fields that we collide at depth in the brain to create an interference pattern. It's called beat frequency. So essentially allows us to stealthily move through the brain at these high frequencies that don't recruit neurofiring, collide them at depth, create a lower frequency envelope that can recruit neurofiring. This was discovered by Ed Boyden's lab at MIT some years ago, validated in a mouse model. Just a pair of Nature Neuroscience papers just came out recently showing for the first time in humans. We've been doing this for years in the lab. We kind of have a glut of papers that are about to be submitted. This is just one of them that we're most proud of, especially this modeling framework that took us many months to finalize, which is kind of raises the bar for how to do non-invasive deep brain modulation in humans. It's an exhaustive search over 100 billion target montage configurations, so it's mathematically optimal. We're searching the entire parameter space. We're finding the optimal montage, and then we're validating it in over 600 individual MRI brains. Happy to talk more about that. That's how we get our protocol. We stress test it in striatum. Decades of work about mysterious beta oscillations in striatum. We have some clues, for example, in Earl Miller's lab at MIT, in monkeys using Granger causality, that striatum talks to prefrontal cortex, particularly at the beta frequency band, when you move from abstract rules to concrete motor accessible routines. We have a instruction-based learning test that's novel, designed by our postdoc Wen, that allows us to isolate this proceduralization process. We go from slow, deliberative learning to fast, procedural, fluent performance. Over 100 people randomized, theta condition, beta condition, left hemisphere, right hemisphere. I'll just go over this cursory way. The upshot is we can stimulate only right beta TI of striatum, enhances learning rate. This is the fingerprint of the proceduralization process using a formal model of a Gaussian mixture model. You can use an HNM as well. And you see the reaction time distributions are cut up into slow and fast. You start slow, you get fast. In that fast cluster, the stimulation enhances it preferentially, not the slow cluster. That's what that plot is showing. We can pinpoint using hierarchical Bayesian change point modeling exactly when in this short bout of learning trials the TI is having its impact. And here we're seeing, you have this speeding, you have this like more implementation of action over trials. The upshot is that beta stimulation, typically it takes you say 5 trials to get really proficient at this task. but we can get you to reach proficiency with under brain stimulation much, much earlier, approaching one-shot learning. This is frequency specific. All the analyses show null effects when we look at the theta frequency TI stimulation of striatum. Nothing in the left hemisphere. This is just right hemisphere specific. This is also highly specific to the experimental task that isolated this proceduralization process. You look at various cognitive measures, affective, perceptual, motivational factors. Nothing changes with stimulation. To get at mechanism, we apply a biophysically grounded model, Nancy Capel's model we're leaning on here. And essentially we show that the beta selectively amplifying the D1 activity in the striatum, and only in a regime where the cortical input from the PFC is weak. So that's like analogous to the early learning stage. This pathway is specific, so beta is not driving D2 neurons, these spiny projection neurons above threshold. Theta is not driving anything. It was specific to the D1 pathway. We also apply algorithmic level, the Frank style algorithmic modeling to go from, how do we go from the D1, D2 pathway information back to behavior? That's what this model affords us, back to reaction time distributions. This is a cortical basal ganglia model, increasing D1. So when we construct the model with D1 dominance, what does the behavior look like? And the upshot is we can reproduce what we find empirically with what the model is finding when we have D1 dominance regime in place. Namely, not just faster learning, but temporal precision to the motor responses is like prototypical learning as well. That is, we apply the same Gaussian mixture model and we get the same selected effect on the fast cluster under D1 dominance. And this is really quick. It's just to some information about why the right lateralization. We in a large, in the CAN data set, over 600 brains, seeded analysis in the right hemisphere and beta, but not theta and not left. And we get a unique connectivity signature of the positive correlations, which we think might help between striatum and cortex, which we think might help explain why the right lateralized effect. There's other reasons too about right hemisphere and striatum has more dopamine concentrations. So this just summarizes what I said. The non-invasive frequency specific control, deep striatal circuits, selectivity of beta TI to shift behavior toward automatic execution, effects that are frequency hemispheric and task specific, the modeling showing us that maybe B1 beta is biasing D1 mediated proceduralization, and that the rhythms act as control parameters for behavior is the conclusion. So the final slide of the talk is that kind of stepping back, I tried to briefly sketch that rhythms are not just correlates of brain activity, but they can act as causal parameters. If we change frequency or phase, we can causally change how circuits communicate, how behavior unfolds. The effects aren't generic. They can be rather specific, bidirectional, in some cases durable. And importantly, the framework scales from these basic cognitive processes across memory and control and learning, symptom-relevant circuits and psychiatry, and across domains. One consistent theme that emerged was that baseline state matters. It can predict who benefits and how much they benefit. And finally, when we move beyond cortex, we can begin to access deeper circuits that likely drive many disorders. And so the opportunity is not just, to stimulate the brain, but to control circuit dynamics in a principled mechanistic way. Thank you.